I also want to mention that I don't have any conflicts of interest because my lab doesn't have money yet. Anyways, the objective for today is to have a bit of an overview of malaria worldwide, discuss conventional approach to diagnostics, some barriers to control and elimination, and how omics method can shed some light on these infection dynamics and help understand malaria better. And we're just going to start with the World Malaria Report 2025, which was released at the end of last year, so very recently. And if you just look at the front picture, you see a rapid diagnostic test, so point of care, near patient testing. You've seen them during COVID, but they are widely used for malaria. We see some lab tests and lab tubes, so again, like laboratory and diagnostics. And if you have the eye for it, there's a DNA double-stranded on the side, so omics and genomics, so interesting. We had the threat of antimalarial resistance, but also diagnostics and access to treatment. If we go on some basic number, roughly half of the world is at risk of malaria. And as of this last report from the WHO, it's now endemic in still 80 countries. That translates to 282 million cases in 2024 and leading to 610,000 deaths. And particularly children, so under 5 years old, that have very higher risk of complication and death. And there are many threats to malaria control and elimination. Drug resistance from the parasites, pitfall in diagnostics, and some health equity issues in accessing diagnostic and treatment. If we look at this map, which is a world map by the WHO, shows which we are on track to malaria elimination and which one are not, with increasing cases. You see in those based on previous reports, malaria elimination. Translated here in this graph that shows on the x-axis years and on the y-axis malaria cases for 1,000 person in the population. For Africa on the left panel and for South America on the right panel, we have the bit of regional and in green the general technical strategy to eliminate malaria by 2023 or to achieve those goals. And I know the x-axis is a bit small, so I put those arrows. This is 2015 made, 2025, which is, last year right now. And we see that we are not on track to control and eliminate malaria worldwide. So we can ask ourselves why, and there's many reasons, and here we are going to approach only some of them, the ones that are rooted in the parasite code. So as a French, I had to brag a bit. So this is a picture of Dr. Lavrin, who got the Nobel Prize for finding malaria on the microscope and making some beautiful drawings. I'm trying to print a big one for my office, but I can't get the resolution right. I'm a bit frustrated. But and we say Plasmodium and we say malaria, but it's a very diverse group of parasites. And you see on this evolutionary phylogenetic tree, the different species that can affect humans or wildlife in this One Health continuum, and especially all those in red show species affecting humans. That we see right here, being the main prevalent species, the most and the one that I'm going to cover more in this talk. So malaria is a parasite. It's transmitted by the female Anopheles mosquito, and infectious bites leads to the release of spores reach the host liver. Bloodstream within the erythrocytes, so the red blood cells, is called an asexual reproduction. And within that part of the cycle, there's two very specific stages of the parasite that interest us. One is those like mature stages can stick deep in the vasculature of the host, and they originate symptoms of malaria, which could be a coma and convulsion when they stick into the brain endothelial vessels, in the placenta, and also trigger because the red cells burst throughout the parasite cycle. But also those ring forms, so those like younger forms in that cycle, are the ones circulating into the bloodstream. And they are the ones that are targeted by most of the diagnostic tools that we have to date to look at malaria. So, as all as Lavrin's drawings from the 20th century, microscopy. So basically you put it on a slide, something staining and looking at it, either on what we call a sigma that side there. And so the microscopist would basically look at it and count and give a number, so the percentage of infected red cells. But the throughput is quite low. It requires expertise and equipment. And you might say, like, it's just a microscope, but for those who are buying lab equipment, a microscope is not nothing. And then you need to pour it properly, maintain it, have the proper oil immersion, clean objectives. It's not easy to implement. Alternative, which if I was giving this talk 10 years ago, besides pregnancy tests, it was not that famous in general, but with COVID, everybody is now quite familiar with rapid diagnostic tests. These are little cartridge where we can put a sample, we get bands depending on which protein from the parasites are detected. So the throughput is quite neat, but it doesn't require too much expertise. I think most of us know the rapid test during COVID. Doesn't require much diagnostics from when I started, where it was not as widespread, and right now where it's heavily implemented. The last option is to detect parasites' DNA. So most of us are familiar with PCR that we can run in labs and that require a fancy thermal cycler. But there are other methods of nucleic acid detection that are based on isothermal amplification, so just one temperature. So one heat block, one temperature. And those methods can be implemented at the point of care to diagnose malaria in endemic areas, but also as screenings in some Canadian hospitals. So the expertise is a bit less than a regular PCR. The equipment is a bit more simple. Throughput is high. However, there are some limitations in those tests. I'm going to discuss that a bit later. And how do those tests perform? Because depending on what the target and the technology behind it, we can see either only if there's a lot of parasites or we need to be less sensitive. When it's based on proteins and detection on the microscope, we can detect up to 50, some people would say 1, depending on the microscope, but parasite per microliter. But with DNA, we can dig very deeper and see those submicroscopic or low-level infections. If we root back into the parasite cycle, usually during the incubation period, the 7 days between the mosquito bag, and we don't see anything. And then the asexual cycle starts and the parasite's level starts to rise. And this is where we can diagnose people with malaria symptoms. However, there is still a bit of a black box in how to assess properly therapeutic efficacy. So how to connect this with what happened in areas where malaria is endemic? We can see on this graph provided by the WHO in their report that rapid diagnostic tests sales have been and use have been rising and completely overtaking the market compared to 2010 to right now, where it's more than 400 million of RDTs used globally. But there is a pitfall to that. Those rapid tests detect a protein called HRP2. And since 2010, there have been reports of deletion of this protein in the genomes of parasites that are endemic, first in South America, and newer reports have also identified this in Africa. So what happens basically is those rapid tests, they will still have the control band because the test works, but the specific protein that is detected from the parasites, the gene being absent, will have false negative tests. So people would present with fever, most likely malaria, but the tests say no, and then they are not likely to access care and treatment. And this has been a shift because if you look at the WHO strategies and the WHO report, if more than 5% of the parasites in the area are resistant, are deleted for HRP2, this country should switch from an HRP2-based rapid diagnostic test to another type of test, which are either less sensitive, less stable, or have some other limitations. This study from Dr. Sindhu Feleke came out in 2021, but it's been very interesting because they blend this point of care approach with some omics method to demonstrate parasites evolve against rapid diagnostic tests. So what did they do? They're conducting surveys on different areas of Ethiopia, so in Eastern Africa, and they looked at the discordance between rapid tests and a PCR testing to look for parasites presence. Each of these tests, other protein detection. They had discordance up to 20 to 30% of the tests. And what I really like about this study, which I think was very smartly designed, is that they the chromosomes in the parasites that carry those HAP2 genes. Particularly here are chromosome 8, so with 2 control strains that are on the bottom here, and they apply 2 omics methods. One being whole genome sequencing, and the other one a molecular semi-targeted sequencing, what they call MIP. And we see here we have the HAP2 gene, deletion in the control strain are present in the other control strain. And it's not just HRP2 that's deleted, it's all the subtelomeric region of the gene, which has very impact. Time it was the largest prospective study, they used multiple molecular approaches, and they demonstrated that this HRP2 deletion in Ethiopia, it emerged from a single origin, but also the HRP2 deletion might provide a better adaptability to the parasites in low intensity settings, and that it was altering the virulence because of those flanking genes in the chromosome 8. So this is really a blend of care to assessing the parasite biology. We can understand what's happening and how it impacts care and patients. A second issue that's happening worldwide in malaria is antimicrobial resistance or antimalarial resistance. So from Pelletier and Caventou using quinine bark in the 1600s to right now our in combination therapy, the parasites developed resistance because they evolve under pressure. And this graph shows the time on the x-axis, those different drugs and their families and combination of drugs that have been used. And every time you see a little thunderbolt, it means this is when the first resistance was documented. It's not very promising on that front, let's say. And if deeper even, and where those mutations associated with resistance were identified, these even areas where they were identified actually in parasites presenting deletion in those HRP2 genes used for diagnostics. It has been shown in different studies in multiple areas, but basically the parasites which are HRP negative, so resistant to diagnostics, also present resistance to artemisinin, which is the most used type of therapy for malaria nowadays. There's been different marker, different approaches, and it's a worrisome situation. So a little bit of malaria in travelers in this context of infection clearance and infection dynamics. And on that front, it's a study we at patient presenting with, and what happens when we follow them up for 3, 7, and 28 days as per the WHO criteria, using blood smear, molecular testing, and rapid tests. And what we've shown is our patients were cured, and we were in France, so there is no risk of reinfection. However, in 12% of patients, 28 days infection, and around 50% 7 days after, the test is still positive, especially those molecular tests. So no local transmission in France. What's happening? Do we have submicroscopic persistence of parasites? Are there residual parasitic DNA and proteins hanging around? And those are really matter, because if we away from rapid diagnostic protein-based and look at genomic testing or like DNA-based assays, we need to know what's happening at that level. We also confirmed this in an independent study, so we still have positive molecular test and HRP2 persistence up to 28 days post-diagnostics. And like malaria is a global issue. We have more positive tests, they are more sensitive, but how does that correlate in our global world and how we are all connected? And I was just curious, because as a French, I'm not allowed to donate blood in Canada because I lived in France during the mad cow disease I'm that old, area. But I was looking up what is it in Canada to donate blood if you've been in endemic areas. And in France, it's a lot. And here, actually, if you look at the website from Canada Blood Services, there is a waiting period up to years when you leave a malaria endemic area. And so far, to my understanding of this criteria, you're not allowed to donate blood if you have had malaria. And it's even addressed on the Canada Blood Services website, where they say it seems disproportionately impacting people from Africa, South Asian, and South American countries. And they say we truly empathize with being turned away from blood donation may leave donors with a sense of frustration and disappointment. In a connected world, and people travel and go around, and we still don't really know what happens long term when you've been cured from malaria and what does happen. So a bit of a topic, but I know it's dear to people in the room here, so I had to talk about it a little bit. So we've discussed that, so malaria is a parasite, it can stick in the vasculature, but in the case of pregnancy, it does something even a bit particular. So again, if we have half of the world in malaria endemic area and half, it means every year there's around 36 million pregnant women at risk of developing malaria. And 13 million of them will end up infected with malaria, resulting in 34% of pregnancy in average having malaria in pregnancy. And the numbers have increased compared to the previous report of the WHO, but it's still only 45% with appropriate prevention. And that prevention is based on using drugs, and some of the parasites are resistant to those drugs. So this is the question to ask. But nonetheless, those infected parasites, they can bind and stick to the placenta, particularly to the chondroitin sulfate A. And this impairs the exchange between the mother and the fetus. It leads to low birth weight newborn or preterm birth, overall negative birth outcomes. And there's also all the pharmacokinetics, like as a pharmacist, anything related to pregnant women pregnant people is always a bit more complicated in how drugs are distributed and cleared. And I've been privileged to participate in a diagnostic outcome study enrolling women during pregnancy and following them after throughout pregnancy and following the newborns after, and implementing molecular testing as a way to pinpoint submicroscopic infection during pregnancy. And long story short, we didn't show any benefit of molecular testing. So those positive PCR tests or positive lab tests, they did not translate in better outcomes. So those people wouldn't have been treated otherwise when they had an asymptomatic submicroscopic infection. We didn't show a benefit. So, yep, I'm going to talk a bit of like, what are those multi-omic approach and how can we have a deeper dive into those host pathogen dynamics? So this time we're going to do this on pediatric malaria, so malaria affecting children under 5 years old. And why are those children particularly sick? It's because the parasite has some multigenic families of genes that confer abilities to bind to different endothelium. So specific ones for pregnancy-associated malaria, some in the brain vasculature, in the lungs, and this is triggering some specific symptoms. And those symptoms are clearly clustered by age, with children having more coma, renal failure, jaundice, acidosis, compared to the adult clinical presentation. So this is a study that was conducted in West Africa, in Benin, and we looked at children under 5 years old, looking at plasma and erythrocytes and using untargeted proteomics approach to try to kind of like decipher what's happening at a more granular level beyond those targeted tests that are used. And what we showed using some like fancy mass spec analysis is that the host plasma proteome reflects malaria infection. We can clearly cluster who is infected, who is not, and if someone is infected with a severe or mild form of the disease. And importantly, we could, through like protein abundance and relative abundance, so here we are presenting some Z-score, so it's a normalized way to look at protein abundance, that children having a mild form of malaria compared to those having a severe form of the disease presents a different overall profile of some plasmatic proteins. So what we show here is that there are more potential biomarker and predictors that can infer infection severity. And those non-targeted omic approach, they allow for more discovery-based science, and it's something very exciting. There's a lot left to do in that realm. This is a study published by a team, I think, from the UK, and they looked at the other side of the story. They looked at the host immune response, and they did some fancy single-cell RNA sequencing. So they took and they disaggregate at the individual cell level what was the expression. And they looked throughout diagnostics and follow up at 7 and 28 days, what were the different genes for type of white cells. And they found very strong evidence of this malaria-induced immunosuppression. And they call it like a multiple tolerogenic response during malaria. And they made all their data publicly available, so as a groundwork for future studies of looking at which parasite variant might be more or less damaging in the long term in this population. Just for the sake of time, because time is running short. I just wanted to say there are some international consortiums that are extremely helpful and funded. For example, the Malaria Gene Consortium has more than 33,000 genomes of Plasmodium falciparum available, and this is really exciting to support different studies. I want to summarize that half of the world right now is at malaria. The parasite biology is very fascinating. If you grow it in the lab, the reason to come on a Sunday night to just look at your smears. There's still a lot to understand, but those parasites, they are a threat and they are evolving. They escape diagnostic. They resist to treatment. So that makes it a hurdle to treat malaria and eliminate it. The research effort to address malaria issue, it's from the entire global health and world health continuum. It's not like one field or another. And I want to thank you for inviting me and your attention. I want to thank people I've been working with before. People who gave me a chance as a new PI to work with me, because it's not easy for trainees to trust somebody new. And I also really wanted to thank Stephanie Sedami and Kim for helping me getting set up with malaria, because as a new investigator, it's not easy. So thank you so much. And giving me a chance of talking here. I hope I can make connections with people here. If you have any questions, I'm happy to answer them. And I'm a heavy coffee drinker. I have nothing against tea drinker. And I really think the best conversation happen around a cup of a warm beverage. So if you want to chat more and connect, shoot me an email. And if there's coffee in the title, you're sure I'll reply very quickly and find time to chat. Thank you again.